Okay, so we'll talk about the modern day phenotype. I will start by providing brief descriptions of what we mean by phenotype and genotype. So phenotype is any observable characteristic of an individual. It can be biochemical, physiological, morphological, etc. And it results from the interaction between the environment and the genotype, and the genotype being the genetic constitution of the individual. There's one particular type of phenotype, the endophenotype. So it's a concept in psychiatry that refers to an internal characteristic that can be objectively measured. So contrary to what could be a diagnosis or a scale for some psychiatric disorder, here you have a quantitative measurement of a brain structure, for example, or a measure of some other biological variable. And then ideally, it's more reliable and should be also more robust than a clinical assessment. So an endophenotype also should be closer to the causative biological process than an external phenotype, like, for example, the diagnosis. So the idea the idea was the fact of studying or trying to find an endophenotype for a psychiatric condition would allow us to have more statistical power and eventually a more simple biological cause than a clinical assessment. And in particular, David Glann and collaborators proposed this idea of a ranking of endophenotype value. So how to determine which endophenotypes from a large spectrum of endophenotypes should be studied first, no? And they provide this very simple equation, which takes into account the heritability of the condition, the heritability of the endophenotype, and we're happy because very often the heritability of brain phenotypes, neuroimaging phenotypes appears to be pretty high, and then their genetic correlation. Okay. The heritability of a phenotype doesn't tell you much about how actually the genome is encoding or influencing the development of the trait that we're studying, and that's it's genetic architecture. So the genetic architecture of a trait refers to the way in which the genome influences the trait and its variability. So, for example, the fact that we have 2 arms with most of the time 5 fingers, but then also the fact that the arms here, the people in the room, they are not all the same length. So it's already the definition of the trait, but also its variability. This is very interesting, in particular for the idea of heritability. Very often people tend to interpret the heritability of a trait as being the fact that it's controlled or not by genes. That's not the case. It's the variability of the trait in a population that may be controlled by inheritable factors. So the genetic architecture of a trait can be described under many different axes. So one is, for example, if the trait is produced by many genes or just a few, so that would be the polygenicity, so the fact that it's dominant or additive, so the fact that the effect of different genes will just add up to produce the final result, or that there would be some, the fact that one allele would, if it's present, it will immediately produce a saturated trait, for example. Then the pleiotropy and the epistasis, so the fact that one gene can affect multiple different traits, or that multiple different genes can interact in a multiplicative way to produce a specific trait. And I will go more into the idea of the polygenicity of genetic architecture. So to caricature the situation, I will illustrate the case of Mendel and Fischer. On one side, so the Mendelian side, we can imagine a trait that's determined by only one allele or just very few of them. On the other extreme, we have the infinitesimal model of Fischer, where eventually all the genome, like every single letter of the genome, has an infinitesimally small effect on the definition of the trait. This can be seen like something like this. Imagine that each of these dots is an allele in our genome. So if you are far enough, you will be able to see the painting of Marilyn Monroe. by Andy Warhol. And then you can see, so what would be some sort of infinitesimal effect of the encoding of the image of Marilyn here would be that, each point is slightly correlated with its neighbors, and it's more the addition of all these points together that produces the trait, which would be the face of Marilyn. On the other extreme, you have this red spot there, which may be, for example, a mutation, so a gene that was lost, a de novo mutation, and it's like very, very focused, and it produces a very specific trait in a specific location. So how do we study these different types of architecture? So one way of looking at the relationship between genotype and phenotype is through genetic association. So genetic association is the systematic co-occurrence of one or more genotypes within a population with a phenotypic trait. Okay, this is the basis of the genome-wide association study that JB described. So, for example, if we look at single nucleotide polymorphism, so this changes in one letter. So most of our genomes are basically the same. So here I have about 1 letters of our genome. So most of them are just exactly identical among all of us. But then, like 1 letter in 1,000 or a few thousands will show a polymorphism. So it may be different between you and me. Like here, a C that can be eventually also a G in some other part of the population. So it's a polymorphism. It's not, most of the time is not associated with any disease, and it's very frequent. So it's a large, substantial part of the variation in the genome. So the difference between individuals would be due to this sort of polymorphism. Now, what we would like to know is if this like frequent variability in our genomes, it's also responsible for the frequent differences that we observe in the traits in a population, like, for example, differences in height or differences in the risk to a specific psychiatric condition, etc. And this is one example of what would look a significant association. So you have one allele, it can have these 3 different genotypes. It can be either AA, AB, or BB, and you observe that more you have a B allele and more the phenotype will increase. Okay, so this is an example of what would be a linear association between the genotype of this allele and the phenotype. Now this is another way of looking at the relationship between genotype and phenotype, and so it's the idea of heritability and genetic correlation. It's very interesting because this is like historically the first way in which we started to assess the effect of the genome into the phenotype of a population. So at a time where we were not able to sequence the genome. So to start with the description. So heritability is, as JB indicated before, the amount of variance in a trait that's due to the genetic similarity among individuals in a population. It's very important to keep in mind that this is relative to a population of subjects. And then the genetic correlation between 2 traits is the proportion of the variance in these 2 traits that's due to genetic causes. Okay, so the fact that, for example, the length of my left arm is determined by the same genes as the length of my right arm, which may not completely be the case, for example, for my head. Of course, there may be a relationship between the size of my arm, the length of my arm, and the size of my head, but there are some other genetic causes that may allow for variation in head size independent of arm size. Okay, so traditionally, the way of looking at this at the time where we were not able to genotype subjects was through, for example, twin studies. We can know if we have monozygotic or dizygotic twins that they will share either 100% of their genome or 50%. Okay, and very often we observe things which look like this. This is the height for 2 twin couples. On the left, you have a couple of monozygotic twins. So the height of twin 1 versus the height of twin 2. And what you observe, if you have seen monozygotic twins, is that very often they tend to look very, very similar. So their heights are strongly correlated. This is not infrequent to observe a correlation of 0.91. On the case of dizygotic twins, which share as much genome as any like brothers and sisters, you observe a correlation, but it's weaker. Okay, so you have the height of twin 1, height of twin 2, and a correlation of often about 0.51. You can derive a very simple formula to estimate the importance of the genetic factors in determining this sort of observation. So first of all, what would be the case if the trait were completely due to environment, like completely random? No, we should observe no correlation at all. Okay, it's interesting also that these twins, because they were born at the same time in the womb of the same mother, they shared a large part of their environment. Okay, so if all the variation in height were due to the fact that we grow in the same environment, the correlation for the monozygotic twins and the dizygotic twins should be the same. So here we observe that that's not the case. There is, of course, a degree of correlation, but the correlation is much higher in the case of the monozygotic twins. And this is the very simple formula. So you can decompose the correlation between a part that would be due to the inheritable factors, so the 100% of the genome that the monozygotic twins share, or the 50% in the case of the dizygotic, and then a part due to the shared environment. That we will suppose, and this is a big assumption, that it's exactly the same for monozygotic twins and dizygotic twins. So in the case of the correlation for monozygotic twins, you can express this as A plus C. And in the case of the dizygotic twins, it would be half of A being the additive genetic factors. And then, so if you just do some algebra there, and then you try to get the value of A, that's also known as H squared, you observe this Falconer's formula, which is twice the difference between the correlations. So in the case that I was showing for height, it would be twice the difference between 0.9, more or less, and 0.5. So twice times 0.4, 80%. And it's more or less the estimation that we have today for the heritability of height. It's 80% due to inheritable genetic factors. You can also derive the value of the common environment, as you see there, and also the value for the random environmental effects which are particular to each individual. And this is the ACE values that you observe very often when people use more sophisticated methods to estimate these variables in, for example, structural equation models. Okay, but the basic idea, I think it's very intuitively seen in this Falconer's formula. Now, as an extension that allows some of the methods that we use today to look at heritability, comes from the fact that you can replace, because now we have access to the genome of very, very large populations. You can, instead of suppose a priori this 1 or 1/2 value for the genetic similarity, you can actually go and compute it. No? You can genotype subjects and then look at what's the percentage of the letters which are different or the same in any pair of potentially unrelated individuals. And so that alpha value can enter the equation as you see at the bottom of the slide. And now these values, they are being used a lot recently. One of the main tools is GCTA, which is a genomic complex trait analysis tool. But more intuitively, to try to understand what this means, you can look at the ancestor of GCTA, which could be the Heisman-Elstrom regression. So the idea is very simple. On the x-axis, if you look at the left, you have A, that would be the genetic distance between subject I and J, and then we are looking at the trait Z, so the phenotype Z, and here you have the phenotype Z for subject I and the phenotype Z for subject J, and this is a Z score, so it's a standardized measurement of the phenotype. And you can simply do a linear regression where you have the multiplication of Zi and Zj, so the traits of this particular pair of individuals. That would be a mean value plus a slope, which gives the heritability plus some noise. You can also use the same idea to compute the genetic correlation. In this case, you will not be multiplying anymore the same trait in both individuals, but you will be multiplying one trait, so trait Y in subject I, and trait Z in subject J. And this time, B will give you the genetic correlation. There are some other methods, but so for example, LD score, if someone wants to discuss about that, I'm very happy to do it, but for the sake of time, I took out the slide. And now I will try to show some of the recent neuroimaging endophenotypes that are used or assume some of these genetic architectures. And it's a very simplified vision of a field that's becoming extremely rich. So this is a first example. It's a twin study. No, it's a pedigree study. So it's not only twins, but there are also some other family members by Anderson Winkler, published in 2009. What Anderson looked was at the surface, thickness, and volume of different brain regions. So the data was extracted from a bit more than 400 subjects, and using free surfer and the standard parcellation of free surfer. What's very interesting is that he observed a substantial heritability of about 0.8 for brain volume and also for the volume of many different regions. He also observed a substantial heritability for the thickness and for the area, but what's interesting is that the genetic correlation between those heritabilities, between those traits, so thickness and area, was very low, which could be interpreted in the sense that the thickness of the cortex and the tangential expansion of the cortical surface area may be supported by different genetic processes. And we know that in one case it would be more the inside-out formation of the neocortex, and in the other it could be more the symmetric divisions of the neuronal progenitors in the ventricular zone, for example. This is another study of heritability based on family relationships. What they were looking at, and collaborators were looking here, was at the genetic correlation between the surface expansion in one point of the cortex and another point of the cortex. Okay, so instead of looking at left arm and right arm, they looked at surface expansion here, surface expansion there. And the surface expansion is the surface expansion of one subject relative to the free surfer atlas. Okay? And what they observed is that there are this set of regions, 12 different regions, where the surface expansion seems to be produced by the same genetic factors. Okay, and you can see on the side the correlation, the genetic correlation matrix. So they observe this series of parcels where the variation is more, it seems to be supported by the same genetic factors. It's very interesting because they made the observation that this sort of parcellation looks similar to the basic parcellation that you can observe, for example, in a mouse brain. So it could be some sort of basic template of the variability in a neocortex, they suggest. Now, this is when we go to actually to the genotype. Okay, so this is the first paper of the Enigma consortium that Jason Stein was the first author. This was probably the first time that our field did the effort of gathering data from a lot of different sources. They had to do an incredible amount of work to try to get a reliably measured phenotype across many, many different sites. And so the phenotypes had to be very easy to compute and reliable to implement in different places. So the first paper looked at intracranial volume and the volume of the hippocampus. So this shows more or less the pipeline that was used for the extraction of intracranial volume and hippocampal volume. So some sites were using FSL, some other sites were using free surfer, and there was even some manual segmentation, if I remember one. And what they observed, although we know that the heritability of intracranial volume and many different structures is pretty high, on the order of like 80% for intracranial volume, the GWAS showed only one significantly associated single nucleotide polymorphism for intracranial volume and only one for hippocampal volume. Okay, and this was basically the time where people were wondering about the missing heritability. This was not only the case for these brain phenotypes, but was also the case for height, for example. We know that height is strongly inheritable, as we just saw, but when people started doing the first GWAS for height, they were able to capture a tiny part of the variability with only a few SNPs strongly associated. Later, Derek, Ibar, and collaborators looked at the volume of subcortical structures in a much larger population. Original population was a bit more than 10,000 subjects, 20,000 subjects for some other phenotypes, and in this case it was more than 30,000 subjects. Okay, these are the subcortical structures that were segmented at that time, and here you can see the Manhattan plots. So showing the significance of each of the SNPs that were studied in the genome for different phenotypes. The results that had been found before for intracranial volume were replicated, and then there was a significant signal also for the putamen. Again, the big effort was to try to get a reliable measurement of the phenotype in a very, very wild, planet-distributed community of researchers. And interestingly also, Derek looked at the shape analysis. So for the allele that was associated with putamen volume, there was a more detailed, sophisticated analysis that was done to try to look what this volume change looked like. So those are normal variation. No? It's the normal variation in putamen volume, the normal variation in intracranial volume. But now our aim would be to try to look again at this idea of the endophenotype. So try to see what would be the relevance of this information for our understanding of psychiatric disorders. And this is pretty much work in progress, but there's some information that's interesting, probably not the one that we would have liked to listen. So this is an analysis of the relationship between the alleles that seem to capture most of the risk to schizophrenia and the alleles that seem to capture most of the risk to the volume of different subcortical structures in particular. So do I have... Okay. So to understand this plot, you have here a guide. So on the x-axis, the SNPs for the risk to schizophrenia were ranked from those that have the higher, that produce the higher effect to the lower effect. And on the y-axis, from those that produce an increase, the largest increase in the volume of the structure and the lower increase. So and here you have a guide. So we have the impression that there is some association between the effects, but actually the statistical analysis show that there's no significant result for the moment. So that first message would be like a note of caution. So pretty much work in progress. But the same paper, and this is like the light in the paper, where we're not completely doomed. It seems that, you know, in the original premise of the endophenotype was the idea that the effects that we would observe are larger in the case of the brain phenotypes than what would be the case for observable phenotypes, like, for example, here, the risk to schizophrenia or variation in height. And this plot seems to show that, in general, the effect sizes, although they are small, as JB indicated, so smaller than 1%, seem to be, on average, larger for brain phenotypes compared with some other observable phenotypes. So there's hope. Now, this is in particular the work that we have been doing in collaboration with JB and people from the Imaging Consortium in particular, so trying to apply these more polygenic sort of methods, the GCTA type of approach. And one of the results that appear from this paper is that it seems that the variation in the volume of different subcortical structures and brain volume and intracranial volume is supported by a very, very large number of SNPs, so which makes our task more difficult. It would be difficult probably to detect specific alleles associated with disease because they may be of very small effect size, something like what would look like the Fisher infinitesimal model. But it seems that if we take all those alleles together, their additive integrated effect is able to explain, nevertheless, a large part of the variance. In this case, with a population of a bit more than 1,700 subjects, we were able to capture about 50% of the variance in many of the phenotypes. And more recently, that was also the case in the paper of Chen collaborators for different, the surface of different neocortical regions, that here with a population of 2,700 subjects, more or less, were able to capture about half of the heritability that's explained by twins, so many times more that we are able to explain when we look at individual alleles. So some closing comments, I wouldn't call these conclusions. Neuroimaging endophenotypes provide a rich level of biological information relevant to psychiatric illness. They are complex, however, and likely related to a large number of rare and frequent genetic mutations. We will see more about this later. And neuroimaging endophenotypes have to be selected so that they allow for the reliable collection of data from a very large population of subjects, and that's probably one of the main lessons of the ENIGMA consortium results. And thank you very much. There are some references here. I will keep adding, and this is a Google slide, so you probably will have access to this, but I will try to keep updating it, and this is the shortened link.
Cool, thanks. Are there any questions for Roberto before he leaves? Yep. Do you want to, you can go ahead to the microphone. There's.
But probably the people at the back will not be able to hear.
And while she's walking up, maybe Derek can come up. You can. Yeah.
Hi.
Thank you.
First of all, very nice talk. I just want to ask one thing. How effective will be considering brain connectivity or delay between regions as a phenotype for the jivas?
There have been some examples. There's an ENIGMA subgroup that's dealing with structural connectivity with diffusion imaging, but also there have been some articles that have used resting state functional connectivity. I think that the main difficulty would be to try to find a way of making agree a large community of researchers into having like similar sort of data. That's very much a challenge for diffusion weighted imaging, and I think that also for resting state, although for example the results of the functional connectome project, if the 1000 connectomes project shows that even if you are pulling data from many different centers, of course there's a big effect, a side effect, but there is nevertheless some common patterns that emerge. So I think that there's hope, but the challenge is very important. If you want to have at the same time the very large populations that are required to obtain reliable results, and not too much methodological noise due to differences in sequence, differences in processing, there's a large effort. There's one initiative inside the ENIGMA consortium that I found very interesting. It's forgot the name of the project, it's a Dutch group, and they are proposing, so very often what has been done up to now is that people upload the results or, here, the idea would be to, instead of moving data, move the researchers. So we are becoming more cheaper to move. So the idea would be to do some sort of a hackathon, and in 3 days, people would go with their hard disk drives to their high-performance computing facility, and then all the data will be pre-processed using the same algorithms, and then at the same time, we would learn how to use the tools, and then I think it's a very neat idea.
That's interesting.
Gather people, do all the analysis in 1 weekend, we will have all our phenotypes.
That's nice. Thank you so much.
You're very welcome.
Any other questions for Roberto? Okay, let's thank him again.